This is the lecture on transcription and translation. It is a very short lecture, and that is reflected in the fact that there are only 3 learning objectives, but this one packs a punch. There's a lot of stuff going on in here. It is not entirely straightforward. I will do the best I can in explaining it in this lecture, but we will do some activities in class that will hopefully make it all make sense and kind of put it all together. But please do let me know if you have questions. Transcription is the process of copying DNA information into an RNA sequence. And the first step is the synthesis of messenger RNA, or mRNA. And that is dependent on an enzyme called RNA polymerase. So why does this need to happen? Why do we need to get DNA into RNA form? And the reason is that DNA stays in the nucleus, okay? It replicates in there and it expresses genes in there, but the protein production part of it happens outside of the nucleus. And you need a way to get the message from the DNA out to the ribosomes to make the protein, and that is messenger RNA. So messenger RNA transcribes DNA's message so that it can leave the nucleus. So the RNA nucleotides here read the DNA strand, and they form complementary base pairs with one of the 2 strands of DNA. So the RNA polymerase recognizes the correct strand and where to start, the start point for the gene, and that always begins with a certain code, and that's AUG, and I'll show you that on a different slide. But here's the DNA strand, and the RNA polymerase is reading this strand and adding RNA nucleotides to the DNA nucleotides to make an RNA strand. Now, important, I said in the last lecture, RNA does not have thymine, it has uracil. So anywhere that there's an A on the DNA strand, there's going to be a U on the RNA strand. So transcription produces mRNA with RNA polymerase. This happens in the nucleus of the cell, okay, and this is the enzyme that does it. The RNA polymerase binds to a promoter sequence, which is always AUG for methionine, okay, it says start here. And the DNA actually unwinds. So here's the blue DNA molecule, and you can see it's unwound by this mRNA polymerase molecule. The mRNA is formed as the RNA nucleotides read this strand of the DNA and come in, and they're kind of strung together. They're attached to each other in one long strand. Now, they don't stay stuck to the DNA. They kind of come out the back end. So here's the newly made RNA being released from the DNA as the RNA polymerase is moving along the strand. I will show you a video of this in class as well that really helps, and it's also helpful to watch the crash course video. The reading stops at a termination signal, and the RNA polymerase releases the new mRNA and leaves the DNA, and the DNA rewinds into its double helix structure. And that is the process of transcription, simply reading the DNA to make a strand of mRNA. Now, when you have your strand of mRNA made, it has something on it called codons. And if you remember what a DNA triplet was, a codon is simply an RNA triplet. So it's a series of 3 bases. So each amino acid in the protein to be synthesized is represented by a series of 3 base sequences called codons. So here you have a few of them. AUG, this is methionine. AUG, and then, well, this is GCA, and I have CGA down here, but that's arginine, okay? UUG, that's leucine. So each 3 base pairs codes for a particular amino acid. So these are all the potential codons in an mRNA strand. You do not need to memorize these. I think it's interesting to see that you can have lots of variations of codons code for a single amino acid. Like all of these code for leucine. All of these code for isoleucine or methionine. All of these code for valine. And the point here is that you can have one nucleotide change or mutation of one nucleotide, and oftentimes you're going to end up with the same amino acid, and that's just kind of like a backup system for your genome to still produce the right protein with the right amino acids, even if you do have single mutations in there. Okay, so now we have our protein code. The mRNA is the protein code. Where does it go? Well, it has to get out to the cytoplasm and get to a ribosome. So it leaves the nucleus through a nuclear pore and goes out and associates with the ribosomes. And now we're going to get into the process of translation, because ribosomes translate the nucleic acid code to the language of amino acids or proteins. Translation, in addition to requiring the mRNA, requires something called tRNA, or transfer RNA. It is a 70 to 80 nucleotide molecule, so just a little nucleic acid, one end of which bonds to an amino acid, and the other end has an anticodon. So it's kind of, I'll show you a picture of them. They're in a structure where one end has an amino acid on it, and the other end has an anticodon. So transfer RNA brings amino acids to their correct place in the chain by binding to codons on the mRNA. So codon, anticodon. Translation also requires ribosomes, as I've said. They provide the enzymatic activity for the bonding of the amino acids and the formation of the polypeptide chain. The polypeptide chain is simply the chain of amino acids, or the primary protein structure. So here are the figures from your book, and again, there are helpful videos on YouTube, and the crash course video is really good. This brown blob is the ribosome, okay? This long strand beneath it is the messenger RNA, and the messenger RNA is divided into codons, okay? 3 bases is a codon. So the 3 bases kind of go through the ribosome, and then you have these tRNAs. So they're actually shaped like a T. One end of the tRNA has an amino acid on it. One end has an anticodon. And if you look, this anticodon is UGC, which corresponds to ACG, which is codon number 5 on the messenger RNA. So the ribosome would start down here, and this is amino acid number 1 for codon number 1, 2 for codon number 2, 3 for codon number 3, and it moves along the messenger RNA, and the transfer RNA molecules bring the amino acids into the right place and join them to the codon that's before them in line. So this 6 will join with 5, and then their job is done and they leave. So here you go. This is the same picture down here. You've got 6 joining with 5 by peptide bond, and then this codon, this transfer RNA that was linked to 5 is done and is gone, and this transfer RNA brings their anticodon down to codon 7, and they will join 6 and 7 together, and the polypeptide chain grows. So transfer RNA brings the correct amino acids into the right place by matching their anticodon to the codons on the mRNA. So these are the steps of protein synthesis, starting from transcription, which makes mRNA, translation makes protein. And that, you have to remember that. Transcription makes mRNA, translation makes protein. Transcription makes mRNA, translation makes protein. And these are the detailed steps for each of those. If you look at the whole picture of everything all together, you have your DNA double helix, the strands get pulled apart, and the RNA polymerase, so the enzyme that makes the mRNA, reads the DNA strand, matches the RNA nucleotides to it, and creates the mRNA. The mRNA leaves the nuclear pore and it attaches to a ribosome in the cytoplasm. The ribosome reads the mRNA, and the transfer RNA molecules come in, match their anticodons to the codons, and join the amino acids by polypeptide bonds. And now what you have is a chain of amino acids, which is the primary protein structure that corresponds to the codons on the mRNA. That's transcription and translation in a nutshell. It's a lot of information in a short period of time. We're going to be doing activities to complete this, to kind of practice it and see what it looks like, get our hands, do some hands-on stuff with it, and we'll be watching some videos in class as well. visualizing this is a really important step to understanding the process. That's it for the lecture.